Right, so the proteins are synthesized on the ribosome, right? So they are synthesized on ribosome. They must fold into their native conformation. But so we'll see a pathway. All right. So for example, this is ribosome. So proteins are synthesized in ribosome. So for example, this is one kind of protein that has been synthesized. So the most that fold into their native conformation, some of the protein that come out from the ribosome, they must fold into their native conformation. But sometimes this occurs spontaneously, and but more often, so the folding occurs spontaneously, but more often, this protein folding requires assistance of some specialized enzyme and complexes. So what the complexes are known? Chaperones. So for example, this is unfolded protein. So this should be folded with the help of this specialized enzyme, that is chaperone. So that will help for folding protein synthesis. Then this protein, unfolded protein, is folded by the help of specialized enzymes, chaperones, and the protein will be folded. All right, so for example, this and this, whatever. So I will put folded protein. All right, so this will be done to folded proteins, or you can say, what you can say, a native protein as well. That's a proper term for that. And this chaperones not only fold the proteins that are newly created, but it also fold the proteins that are unfolded during the any reactions or interaction. If some proteins are unfolded, it will, this chaperones also help for unfolding. Right, so after this native protein is created, so for example, it goes to some reactions or whatever, and then it will become, if unfolded, right, or misfolded or unfolded, folded, and then it should be go to degradation, right, and there will be a peptide fragments, just fragments, or this misfolded protein, if it's required, it would be again folded by these chaperones, right? Or what can happen, this misfolded protein, if it's not degraded or denaturation, then what happens, it can aggregate as well in the cell, and that is dangerous. So the proteins that are not folded properly, right? So for example, this is a native protein, right? And if this native protein is not folded properly by this enzyme, so what happens, the hydrophobic surface in the interior of the protein, they might, what, it might be exposed, and it can be sticky, leading to the formation of inactive aggregates. So these aggregates are inactive, they are not active aggregate for the protein, so they are not helpful. So these aggregates may lack their normal functions, right? So their accumulation in cell are the heart of the disease. So this, if it lies, so they are the cause of some disease. So for example, diabetes to Parkinson disease and Alzheimer disease as well, right? So this aggregate, like the, for example, this aggregates like amorphous aggregate. There is amorphous aggregates. There are oligomers as well, oligomers. And there is the one that you know, amyloid fibrils, right? All right, so this was the proteostasis and how it affects our body. And there is also loss of protein structure results in loss of function. So the protein structures have evolved to the function in particularly cellular environments. So the conditions different from those in the cell can result in the protein structural changes, large and small. So a loss, okay, Write down here. A loss of three-dimensional structures is sufficient to cause loss of function, right? Loss of function, and what's called is called denaturation. So this is called denaturation. So a loss of three-dimensional structure that is sufficient to cause the loss of function is called denaturation. So the denaturated state does not necessarily equate with the complete unfolding of the protein and randomization of conformation. So under the most condition, denaturated proteins exist instead of a prime partially folded states. So the most proteins can be denatured by heat, which has a complex effects on many weak interactions in a protein, that is, and also primarily to the hydrogen bonds. So if the temperature is increased slowly, a protein conformation generally remains intact until an abrupt loss of structure and function occurs over a narrow temperature range. So the abruptness of the change suggests that the unfolding is the cooperative process, right? Because the loss of structure in one part of the protein destabilizes the other parts, right? So the protein denaturated can be affected by the temperature or by the heat. Then protein can also be denaturated by the extremes of pH, by certain miscible organic solvents also, such as alcohol or acetane, or by certain solutes such as urea and guanidine hydrochloride, or by detergents. So these are also few of them that can denaturate the proteins, and each of these denaturating agents represents a relatively mild treatment in the sense that no covalent bonds in the polypeptide chains are broken. So in this, no covalent bonds are broken. Like for example, organic solvents, urea and detergent act primarily by disrupting the hydrophobic aggregation of the nonpolar amino acid side chain that produces the stable core of globular protein. Urea also disrupt the hydrogen bonds, and the extremes of pH alter the net charge on a protein, causing electrostatic repulsion and disruption of some hydrogen bonding. Right? So the denaturated structure resulting from this various treatment are not necessarily the same. Okay, and the denaturation often leads to protein precipitation, a consequence of protein aggregate that we saw here. Formation as exposed the hydrophobic surface associate. Alright, so and there is the tertiary structure of a globular protein is determined by its amino acid sequence. So that means amino acid sequence determines the tertiary structure. So the most important proof of this came from the experiment that shows the denaturation of some protein is reversible. So that means certain globular proteins denaturated by heat or extremes of pH or denaturing reagents will regain their native structure and their biological, even the biological functions activity, if returned to the condition in which the native conformation is stable, and that process is known as renaturation, because the protein is denatured first and then it's again gone to its stable state or the native conformation that is stable. So that process is called renaturation, and there is an example that is done for this denaturation. Renaturation is of ribonuclease. Example is ribonuclease, right? So this is demonstrated by Christian and Finsen in 1950. So what he did, he denatured this completely in the concentrated urea solution in the presence of reducing agent, and this example showed how it is renatured as well. So the prediction of protein structure is especially the field of bioinformatics, and the progress in this area is monitored with a biennial test that is called CASP. That is called CASP. That means critical assessment of structural predictions computation. Right? Alright, so we studied about the chaperones. So there are two types of chaperones actually. So that is the two major families that has been founded or well studied in the chaperones. The first is HSP70 and the second is chaperones, right? So the HSP70 family of proteins generally have a molecular weight nearly 70,000. I hope you can see that 70,000, right? And are more abundant in cells that are stressed by elevated temperatures, right? So HSP70 proteins, what it does is binds to the region of unfolded protein polypeptide or a polypeptide. It's called polypeptide rather than the protein, right? The unfolded polypeptide that are rich in hydrophobic residues. So this chaperones thus protects both proteins subject to denaturation by heat and also the new peptide molecule being synthesized. So the HSP70 protein also block the folding of the certain proteins that must remain unfolded until they have been translocated across the membrane. So the HSP70 protein bind to and release the polypeptide in a cycle that uses the energy from the ATP hydrolysis and involve several other proteins. And the chaperones are elaborate protein complexes required for the folding of some cellular proteins that do not fold spontaneously. For example, in E. coli, an estimated 10 to 15% of cellular proteins require the residence chaperoning system that is called GROEL or GROES, right? Oops. It was just an assay. GROES, right? So the analogous chaperoning system in the eukaryote is also called HSP60. The chaperones first became known when they were found to be necessary for the growth of certain bacterial viruses. Right, and on your screen you can see the system. So what it does, the chaperoning first became. So what it happens, this chaperoning protein is structured as a series of multi-subunit wings forming two chambers oriented back to back. And what it does, an unfolded protein is first bound to an exposed hydrophobic surface near the apical end of one of the GROEL chamber. Then the protein is first trapped within the chamber when it is kept transiently by the GROS lead. So the GROL undergoes substantial conformational changes coupled to the slow ATP hydrolysis, which also regulates the binding and the release of GROES, that's grows, right? And inside the chamber, protein has about 10 seconds to fold, the time required for the bound ATP to hydrolyze, and the constraining a protein within the chamber prevents inappropriate protein aggregation and also restrict the conformational space that a polypeptide chain can explore as it folds. So the protein is released when the GRES cap dissociates and but can rebind rapidly for another round if folding has not been completed. So the two chambers in the GREL complex alternate in binding and releasing unfolded polypeptide substrate. Correct? Alright, so these are some different kinds of folding processes that are done in the cell. So like the folding of proteins is really important. Why it's important? Because defect in protein folding provides the molecular base for a wide range of genetic disorders. So despite this many processes that assist in the protein folding, misfolding does occur. All right, and in some cases, so in fact the protein misfolding is a substantial problem in all the cells, and a quarter or more of all the polypeptides synthesized may be destroyed because they do not fold correctly, right? So if the protein is not folded correctly, it will be destroyed, but in some cases the misfolding causes or contributes to the development of serious disease. So many conditions like type 2 diabetes, Alzheimer's disease, Huntington disease, Parkinson disease are associated with the misfolding mechanism. What is that? Is a soluble protein that is normally secreted from the cell is secreted in a misfolded state and converted into an insoluble extracellular amyloid fiber, and the disease is collectively known as amyloidosis, right? So what happens is that the soluble protein that is secreted from the cell is secreted in a misfolded state, right? Instead of this folded, it's in the misfolded state, and it's converted into an insoluble extracellular amyloid fiber.